Hello everyone. Thank you for joining webinar today. I'm Sonali Jagg, a technical support scientist here at ACD. Before we begin, I would like to let everyone know that the audience will be muted for the duration of the webinar. If you have a question, please use the chat function to the right of the WebEx window. Please address all your questions to all panelists. My colleagues will address your questions in real time. In the event we have not addressed your question, we'll follow up with you offline after the webinar. Please note that the webinar is being recorded. So in the event you cannot stay for the whole duration of the webinar, we will have the recording up on our website. With that said, I would like to begin with our webinar for today. Here is a quick overview of the topics we will be covering today. I will begin with an overview of our RNAscope technology, followed by the general workflow and troubleshooting guidelines. I will briefly discuss interpretation of the RNAscope results. Next, I will discuss BaseScope assays and applications of our technology. Please note that during the duration of the webinar, everyone will have an opportunity to ask questions to our panelists. Towards the end, we will also go over a few frequently asked questions. So let's start today's discussion by understanding what is RNAscope. The RNAscope assay is an in situ hybridization technique that utilizes a unique double-Z target probe design followed by signal amplification system, which allows you to visualize a single mRNA transcript as a punctate dot through either chromogenic or fluorescent detection. This highly specialized system utilizes simultaneous signal amplification and background suppression methodology. RNAscope is compatible with a wide range of sample types such as FFPE, fixed frozen, as well as fresh frozen. RNAscope can be used to detect multiple mRNA targets using one tissue section. The growth and adoption of the RNAscope technology is best exemplified by the number of peer-reviewed publications in the fields such as cancer research, neuroscience, and infectious diseases, just to name a few. We had our first publication in 2011, and since then, there have been over 1,800 peer-reviewed papers published using RNAscope technology in numerous journals, including many top-tier ones. Here is a brief overview of the RNAscope workflow. RNAscope assay is a slide-based assay and is intended for use on sample types similar to that used with IHC or IF. The assay starts with permeabilizing the sample with reagents provided in the pretreatment kit. The pretreatment allows unmasking of the mRNA targets, thus allowing the target probe to penetrate into the cell and hybridize the target RNA sequence. The target probe is a pool of oligos that are designed using our established bioinformatics algorithm, which allows for target specificity. A series of amplification cascades can then amplify the signal, and RNA molecules can be visualized under the microscope as punctate dots. The signal can also be quantified semi-quantitatively or quantitatively using image analysis software. The primary feature of our RNAscope technology is the probe design. Represented here are the oligonucleotide target specific probes as double Zs. They have two unique regions connected by a linker. Additionally, each of these oligonucleotide sequences have been designed using an informatics algorithm which selects sequences that specifically bind to the target sequence of interest. This design confirms that the oligonucleotide sequences do not cross-hybridize with any other sequences. The sequence at the bottom of the Z is complementary to the target transcript and hence hybridizes. In order for amplification to occur, two Zs must hybridize to the target sequence right next to each other. Once this happens, it creates a 50-base target-specific binding site on the bottom of the ZZ pair. The top of the Z is the base for the amplification structure. When two Zs hybridize, it creates a binding site upon which a pre-amplifier can bind and the amplification tree can be built. A standard RNAscope probe for a target sequence of about 1,000 bases or more consists of about 20 ZZ pairs pulled together that are designed to hybridize next to each other along a target region. This allows for a tremendous amount of amplification and signal potential. However, only a few ZZ pairs are needed to bind to the target RNA sequence to generate enough signal for molecular detection. Here is a schematic showing the hybridization and the subsequent amplification of RNAscope assay. The double Z probe will bind to the target mRNA transcript. Once a double Z probe binds, it serves as a foundation and binding site for the pre-amplifier molecule. Once the pre-amplifier is bound, it serves as a binding site for the amplifier molecules. And each amplifier can further bind multiple labeled probes, sequentially hybridizing to assemble a branching complex at each ZZ binding site. Labeled probes can contain chromogenic enzyme such as HRP that generates a visible signal after chromogenic reaction such as DAB, detectable under standard bright field microscope. The labeled probes can also contain fluorophores that allow for visualization of the signal under fluorescence microscope. The signal amplification strategy provides for visualization of target as single dot, where each dot represents an individual RNA molecule. If a single Z binds to the target mRNA transcript, the pre-amplifier cannot bind as it needs both Zs to provide a binding site. This helps to ensure the specificity of the probe and helps reduce potential background staining. In order to detect multiple mRNAs simultaneously, multiple amplification trees must be built. That being said, we must ensure specificity. Specificity is ensured by adding a unique tail sequence to the probes. This means that the double Z probe design will have a tail sequence that is indicative of a channel designator. For example, a C1 probe has a specific C1 tail sequence that will only allow for binding of subsequent C1 amplifier and labeled probe molecules. Similarly, a Situ probe will have a separate and unique tail sequence. This slide presents all the assays that are currently available from ACD. If you have a single-ish target, you may choose our Singleplex assays such as Chromogenic brown or red assay. If you have two targets, you may choose Chromogenic Duplex assay. Both of these assays are available on manual and automated platform. If you are interested in multiplexing, our multiplex fluorescence assay allows to detect up to 4 targets simultaneously in a single tissue section. On the other hand, if the target of interest is shorter than 300 base pair or circular RNA, we recommend the BaseScope assay. I will be giving a detailed overview of the BaseScope assay in later slides. As depicted in the previous table, some of these assays are available on either Leica Bond RX or Ventana Discovery automated platforms. Recently, we have launched Ventana Universal HRP AP assays, which will replace the Ventana RNAscope 2.5 HD Chromogenic assays. If you are interested in the automation platform, please contact your local account manager. We will have our local field application specialists available for training and troubleshooting on the automation platform.
Sure.
In the next few slides, I'll discuss the RNAscope amplification and detection systems for the manual assays. The diagrams here depict the Singleplex amplification system. Pictured here is the amplification tree, which is then tethered by an enzyme. To the left is the depiction of the red assay, which utilizes alkaline phosphatase and can then be visualized by fast red chromogen. Likewise, for the brown assay shown on the right, utilizes HRP and is visualized by DAP chromogen. Brief overview of duplex amplification is presented here. Most probe channels have independent amplification trees. The channel 2 associated amplification tree utilizes alkaline phosphatase enzyme and fast red substrate for visualization of the marker. A separate amplification tree is built for channel 1, which utilizes HRP enzyme and green substrate to visualize the second marker. Please note that our C1 probes by default are ready to use and 1x in concentration, while the C2 probes are provided as 50x stacks. In order to make the duplex probe mixture, combine the C2 probe with the C1 probe in 1:50 dilution. If you would like to test C2 probes alone, please use the RNAscope probe diluent and combine at 1:50 dilution. The multiplex fluorescent V1 assay utilizes the same amplification system and is similar to the duplex chromogenic assay. Here, 3 independent amplification trees are built, such that C1, C2, and C3 probes will have their own amplification trees stemming from where the probe has bound to mRNA transcript. The signal can then be visualized from the 3 independent fluorescent label probes. The fluorescent multiplex V2 assay can be used to detect up to 4 mRNA targets. Here, 4 independent amplification trees are built, such that the C1, C2, C3, and C4 probes will have their own amplification trees built from the mRNA target. Signal can then be visualized by using Perkin-Elmer's PSA-based amplification technology or the Opal dyes. Next, moving on to the RNAscope assay workflow. With the RNAscope assays, you may perform the assay as a one-day assay. If you would like, there are a couple of options to split this protocol into 2 days. The first option would be to complete the sample preparation and pretreatment on day 1 and then begin with probe hybridization and detection on the following day. Please do refer to the technical notes recommended for your sample type to determine where the stopping point may be feasible. If unsure, please do contact technical support for recommendations. The second option of splitting into a 2-day assay would be to complete sample preparation, pretreatment, and probe hybridization on the first day, and then incubate the slides overnight in 5x SSC solution. The following day, you would remove the slides from 5x SSC and wash with wash buffer, then follow with amplification reagents. This is highly recommended with the Duplex assay and Multiplex fluorescent V2 assays, as these are much longer assays with several more amplification steps involved. This is an overview of the workflow for Singleplex chromogenic assay using FFPE samples. The Singleplex assay can take about 8 hours. Please refer to Part 1 of the user manual for sample preparation and pretreatment recommendations. The assay workflow has 4 major steps: pretreatment, hybridization, amplification, and finally staining and detection. The workflow for FFPE and fixed frozen samples require a heat-mediated target retrieval step followed by protease digestion for pretreatment. The workflow on the Leica Bond RX and Ventana systems are very similar to the manual assay. Similarly, fluorescent multiplex V1 assay contains the 4 major parts, but it is a shorter assay. Again, please refer to the user manual for detailed recommendations. Please note that you need to choose only one AMP4 alt option among the ones recommended in the user manual. Just a quick note that if everyone could mute their microphones, that would help in listening to the webinar. So please make sure that your microphones are muted to avoid any background noise. Thank you. So continuing on to the RNAscope multiplex V2 assay. Here is a more detailed workflow for the multiplex fluorescent V2 assay. Similar to the previous workflow diagram, pretreatment is completed first, followed by the target probe hybridization and subsequent amplification steps. This differs from previously mentioned fluorescence assay as each channel is developed sequentially. The AMP1, 2, and 3 steps are a pooled combination of pre-amplifier and amplifier molecules so that the amplification tree begins to form simultaneously. This schematic shown here uses fluorescein, Psi3, and Psi5 in C1, C2, and C3 respectively. But these may be mixed and matched as per your preference. Please note that this is a TSA-based assay and requires the purchase of TSA plus fluorophores or opal dyes from PerkinElmer. Each kit configuration includes pre-treatment reagents, detection kit, as well as wash buffer. Presented here are some recommended accessories required for running the manual RNAscope and BaseScope assays. At the top left are 2 options for washing and handling slides. You may want to use either TissueTech wash tray, common in most of the labs, or you can use our Easy Batch slide processing system, which was designed to streamline your RNAscope and BaseScope assays and save handling time when working with large number of samples. The Hybridization oven, shown on the top right, is designed to maintain the optimal temperature and humidity to ensure optimal and consistent RNAscope or BaseScope staining. Below are the two methods for carrying out heat-mediated target retrieval step, which is required for some sample types such as FFPE or fixed frozen. Our website has training videos on different steps of manual assays, which is a great visual guide to assist with running the RNAscope manual workflow. Some general tips to ensure success with the RNAscope and BaseScope assays are shown on this slide. First, follow the protocols. We have various protocols with necessary details to help address the best way to run the assay. Next, sample pretreatment is very important to ensure that you get the most optimal RNAscope signal. The pretreatment conditions will differ from sample type to sample type and may require some optimization. Using controls is highly recommended, especially when you are troubleshooting your assay. And lastly, it is critical to make sure that you have all the components required to run the assay before you begin your assay. Here is an example of recommended sample preparation. With FFPE samples, we recommend fixing with 10% NBF for 16 to 32 hours at room temperature. The recommended sample thickness is 4 to 6 microns, and we also recommend that the tissue is mounted onto Superfrost Plus slides. In order to preserve RNA quality, it is recommended to store sections with desiccants at room temperature for up to 3 months. For long-term storage, it is best to store at 4 degrees with desiccant. For fresh frozen samples, please review the user manual for detailed recommendations. A few highlights are presented on this slide. We recommend storing frozen tissue blocks or sections in airtight containers at -80 for up to 3 months. The recommended sample thickness is 10 to 20 microns, again mounted on Superfrost Plus slides. Besides preparing samples, according to our recommended protocol, it is very important to have proper controls for RNAscope assay. If you are running RNAscope assay for the very first time, we recommend running positive and negative control probes on our control slides such as HeLa in order to ensure that the assay setup in the lab is optimal. Then proceed running the assay using your control probes on the sample to assess the sample RNA quality and to determine the best pretreatment conditions based on the signal detected as well as tissue morphology. If your sample passes QC, you can then move on to running your target probe. Otherwise, it is recommended that you verify your technique, check RNA quality, or perform assay optimization. I'll be discussing our scoring guidelines in a later slide. On this checklist are some other key components that can help ensure your success with RNAscope assay. For example, Superfrost Plus slides are highly recommended for the success. Also, using Hybridization oven is a critical component for maintaining the right amount of humidity. We also have some recommended general guidelines, such as once you begin your assay, do not let the slides dry out, or make sure that you warm the probes and wash buffer at 40 degrees to avoid any precipitation. When running the RNAscope or BaseScope assays, here are a few factors that could affect the outcome of your results. For example, fixation conditions are not optimal, or hybridization conditions are not optimal. These factors and issues can be addressed by the following solutions, such as the ones that are listed here. Please fix the samples as recommended using the in the user manual, or use the HiBusy hybridization system for optimal hybridization conditions. In the next few slides, I will be discussing some of the key guidelines pertaining to each of the manual assays. Chromogenic brown assay is one of our most robust assays. It is an ideal assay for tissues that have alkaline phosphatase-dependent background, such as kidney samples. But this assay is not ideal when using tissues that are subject to pigmentation, such as lung or skin samples. The fast red chromogen is used in both the RNAscope red assay and BaseScope assay. We recommend using either eco-mount or vector mount media to help best preserve the staining. The chromogen is sensitive to alcohol, and if exposed, may cause signal diffusion. After chromogen development, the slides should not go through ethanol dehydration, but instead, we recommend drying slides in a drying oven at 60 degrees. In the chromogenic duplex assay, your C1 probe will be visualized in green and your C2 probe will be visualized in red. A few critical points to note here are the sensitivity of both the red and green chromogen to alcohol. Therefore, these should not be dehydrated using alcohol prior to mounting. Additionally, we would recommend you to consider splitting this assay in 2 days due to the length of the time. For the fluorescent V1 assay, you can use different AMP4 alternatives A, B, or C that are shown on this slide. For different fluorophore combinations, we recommend using prolonged gold anti-fade mounting media to preserve fluorescence. The multiplex fluorescent V2 assay is a TSA-based assay. TSA plus fluorophores or opal dyes have to be purchased separately from per-kilonmer. Again, we recommend using prolonged gold anti-fade as the mounting medium. Now the next question is, which fluorescence assay would be right for you? The first assay, fluorescence multiplex V1, is best used with fresh frozen samples and consists of labeled probes conjugated to fluorophores. This assay allows for triplexing capability of 3 mRNA markers simultaneously in your tissue or cells. The second assay, multiplex fluorescent V2 assay, is TSA-based assay, where it utilizes Per-Kilmer TSA plus fluorophores or opal dyes. It was developed with FFPE tissues in mind to help address the auto-fluorescence issue. This assay can also be followed with immunofluorescence. We have a technical note up on our support web page for dual ISH-IF protocol. With the additional purchase of 4-plex auxiliary kit, the assay will allow for 4-plex capability to detect 4 mRNA markers simultaneously. The LS multiplex assay is similar to V2 assay, which also utilizes TSA plus fluorophores or opal dyes, but is completely automated on LicaBond RX platform. Now that we have learned about the technology and assay workflow, next we will discuss how we would analyze the assay results. Some of the questions that you may have in your mind at this point are, what does a dot mean? Or what is the significance of the dot size? And what is the difference between a dot and a cluster? So let's take a look at that. In this image, the RNAscope chromogenic assay is shown. This assay was performed using a red chromogen, where signal appears as numerous punctate red dots of various sizes and intensities, and few clusters. It is the number of dots and/or clusters that are important rather than the size of the dot or the intensity of the dot. Each punctate dot equals 1 mRNA molecule. The small faint punctate dot pointed by a blue arrow is 1 mRNA molecule. The slightly larger, darker, nicely round punctate dot pointed by a yellow arrow is also 1 mRNA molecule. However, it is larger in size as more double Z probes have bound to the target molecule. In contrast, a cluster pointed by the brown arrow is more oblong with slightly irregular border and results from overlapping signals from multiple mRNA molecules. Similar patterns of dots and clusters are seen in fluorescent versions of the assay. The yellow arrows point to single mRNA molecules with less ZZ probe pairs bound to them compared to the dot pointed by teal arrow. The white arrow is pointing to the cluster with an irregular rather than round shape, resulting from overlapping signals from multiple mRNA molecules. RNAscope staining can be quantified by manually counting the number of dots per cell. It is also known as semi-quantitative scoring method. With this method, a score of 0 to 4 is assigned based on average number of dots per cell. For more quantitative approaches to the data analysis of RNAscope results, visit the link below where you can download a free guide describing the different methodologies available to quantify your results. If you have further questions, please contact technical support for recommendations. Before we move forward, I would like to reiterate that it is critical to qualify your samples using control probes before running the assay using the target probes.
No, but we can close that. It's just this RNAsec. No.
So why optimize your assay? Here are some of the potential reasons why you may need to optimize. If the samples are underfixed, this can usually lead to over-digestion of your samples using the standard conditions. If the samples are overfixed, the signal could be weak and you may need to optimize. There are also some particular sample types that could require different pretreatment conditions and optimizations, with a few listed here. If your tissue of interest is not listed, or you're unsure if your tissue has been fixed as per our recommendations, please feel free to reach out to our technical support group.
This is important for you.
Sample pretreatment is a very critical step for the success of RNAscope. When running RNAscope assay on your samples, begin with recommended standard conditions for your sample type. Observe for signal intensity and background with control probes and sample morphology.
So I don't know what.
If the recommendations for your sample type are not available in the user manual or a tech note, it is always best to start with standard conditions. This will help to set a baseline for how the assay performs, and based on your results, we can make recommendations to either scale the pretreatment up or down. Both the target retrieval and protease digestion perform on single sliding scale, such that you may adjust it depending on what your samples look like or the fixation conditions of your samples.
Yeah, because or you find yourself.
We'll just pause for a few seconds to mute the audience in case somebody is still not muted. So just please, I'll put on brief hold. Pause for a few seconds. My apologies for that.
They are eating also.
Okay, so let's get started again. So moving on to the next step, which is the sample pre-treatment optimization. Optimization on the automated platform is similar to manual assays. On the automation platform, you would change the epitope retrieval time and temperature as well as time spent with the protease. Provided that the target is expressed in the sample and the protocol is followed as recommended, the RNAscope assay is guaranteed. Because tissue samples can be highly variable, proper sample preparation and the right controls are essential to an RNAscope experiment. Tissue samples must be properly fixed and prepared to ensure good RNA quality and no background due to poor fixation. ACD provides guidelines on how tissues should be fixed for optimal performance of the RNAscope assay. Here are serial sections from human lung cancer sample on which we performed the manual chromogenic red assay. On the left is the section stained with the negative control probe DAP-B, which is a bacterial gene that is not expressed in most tissues. As observed in this sample, there should not be any signal from DAP-B. Next, let's look at the panel in the center. This panel has the sample stained with our positive control probe for the housekeeping gene PPIB. As observed with this sample, you see fairly uniform detection with the positive control probe, indicating that the RNA quality is good. Certain tissues may express the positive control probe at a higher or lower level, but in general, you want to see expression throughout the sample. Now that we have looked at the positive and the negative control probes, let's move on to the target probe. With the results from these two probes, we can have confidence that our test data, what we see, is accurate. As shown in the example, we are looking at the lung cancer sample probe for immune checkpoint marker, which is PD-L1. PD-L1 exhibits a wide range of expression in tumor tissues. In this human lung cancer sample, we observed strong punctate dots with PD-L1 probe, indicating that the PD-L1 expression is present in this tumor sample. The controls show us that the RNA quality is good throughout the tissue sample and that there is very little to no background. And hence, we can be confident that the localized pattern of the expression is as we would expect. Now let's review some of the troubleshooting examples and the results after performing the optimization steps on these samples. On this slide, we see optimal digestion with a tissue fixed with our recommended protocol and then treating with standard conditions. PPIB staining is strong while DAP-B negative control is clean. Tissue morphology is intact and hematoxylin staining is homogeneous. Note that this assay was run on LicaBond RX platform, so the pre-treatment conditions shown are with regards to running this on automated platform. These are standard conditions on the Bond RX. As compared to the last slide, this data set shows that the samples have been over-digested, in which the nuclei are now undefined due to suboptimal tissue morphology, and the PPIB staining is slightly weaker than expected. Again, please note that these conditions were done on LicaBond RX platform. Now let's review some of the troubleshooting examples and the results after performing optimization on these samples. Here, the target retrieval time was reduced to 8 minutes, and that improved the morphology and eliminated background as seen in the negative control. As compared to the image on the left, there is better morphology and better signal resolution on using modified target retrieval buffer or target retrieval time. In the opposite direction, here are samples that have been under-digested. The PPIB staining is much weaker, but tissue morphology looks great, showing no signs of over-digestion. Here, the target retrieval time was increased to 30 minutes, and that improved signal intensity and resolution, and also eliminated trapping of the chromogen, which is seen in the image on the left. Shown here to the right is an image of RNAscope performed on a sample that was fixed for too long. And as you can see, the signal is very weak. And hence, we highly recommend fixing samples as per our recommendations. When the multiplex fluorescent RNAscope assays are successful, you should see images like the ones shown here. This is using AMP4 Alt-A option and was performed on HeLa control cell pellet with the human 3-plex positive control probes. Several customers use fresh frozen samples. The images here display how protease treatment can impact RNAscope signal quality. With mouse kidney samples shown in the top panels, you can see that there is quite a bit of tissue autofluorescence. But when the sample is appropriately digested using protease 4 at room temperature for 30 minutes, distinct green and red dots can be observed with control probes. When protease digestion is performed at 40 degrees, the tissue is over-digested. And not only you do not see any punctate signal, you also see increase in the background. Similarly, as shown in the bottom panels, with adequately treated fresh frozen mouse brain samples, there is no background, and you can observe beautiful punctate signal with tissue. And with tissue over-digestion, you see an increase in background and decrease in signal. One known issue with fluorescence assay is that due to long fixation time, FFPE samples are prone to inherent autofluorescence, which poses challenges for image analysis. You can use FFPE if you have background correction softwares to reduce autofluorescence, and alternatively, we have now developed a multiplex fluorescence version 2 assay that helps to address this issue. If you are still experiencing autofluorescence issues with your assay, please do contact technical support. We recommend SuperFloss Plus slides to ensure optimal sample adherence. If you observe tissue detachment, the likely causes are listed here. We have found that adding an additional baking step can mitigate this issue. I will review the additional baking steps incorporated for FFPE and fixed frozen samples. These apply to all assay types. If you are observing detachment after hydrogen peroxide or target retrieval, then adding a baking step before hydrogen peroxide. Add a baking step before hydrogen peroxide. Or if you are observing tissue coming off from the slides after protease treatment, add an additional baking step before that. Both baking steps are for 30 minutes at 60 degrees in a circulating dry oven. For sample detachment with fixed frozen tissues, we recommend to dry slides at -20 for 2 hours after cryosectioning. And also here adding a post-fixation step before hydrogen peroxide and target retrieval for 15 minutes to 2 hours at room temperature. We have found that majority of sample detachment happens after target retrieval. So we recommend adding additional baking steps before protease step. After trying the above steps, 1, 2, and 3, as shown in the slide, if you are still observing detachment, we recommend you omit the target retrieval step and extend the protease time to 45 minutes. Also, please contact support and we can help you figure out the best troubleshooting path for this issue. For probe-related troubleshooting, if you are working with the 2 scenarios listed here, please contact technical support. With knockout validations, as we have thousands of probes currently in the inventory, we would like to help ensure the probe you are picking is specific for your wild type or knockout validation. If you are working with novel gene and no catalog probe is available, please feel free to contact your area account manager or go to our website to get the custom probe design feasibility test started. We have recommended sample preparation protocols for commonly used sample types. So if you're working with unique samples such as whole mount, free-floating, or bone, feel free to contact tech support for suggestions and guidance. Presented here are some of the key areas where RNAscope has been increasingly used. In the next few slides, we'll go over some of the examples of these applications. For many targets, reliable antibodies for IHC detection are not available. An example of this is LGR5, which is an intestinal stem cell marker. In this study, authors used RNAscope to detect LGR5 in human colorectal cancer samples. LGR5 is known to be a stem cell marker in the murine, small intestine, and colon. However, the localization in human adenoma samples has not been examined in detail. Previous slides in the previous studies due to lack of specific antibodies. Authors approached very systematically to see the localization of the LGR5 expressing cells and to understand differences or alterations in stem cell architecture in human normal and adenoma samples of all stages. For many GPCRs, there are either no antibodies available or no reliable antibodies available. So the detection of GPCRs in the tissue context can be limiting. Dopamine receptors are implicated in many neurological processes. Here we demonstrate detection of GPCRs DRD1 shown in red and DRD2 shown in green using RNAscope multiplex fluorescence assay. Here is an image of bGlut1, bGlut2, and bGAT receptors in FFPE mouse brain using the multiplex fluorescent V2 assay. Here is a high magnified image of the same markers in the mouse brain. One of the most asked questions is, can you combine RNAscope-ISH with IHC or IF? Both ISH and IHC procedures do have similar workflow as shown here. Both require sample fixation, some kind of pre-treatment to unmask your RNA or protein, and subsequent detection. The recommended workflow is shown here, which would include pre-treat, permeabilize, pre-treat or permeabilize your tissue or cells first. Depending on the tissue, this may require both heat-mediated target retrieval as well as protease digestion. You would then complete the RNAscope-ish portion and then follow the IHC staining after that. We do have some recommendations to ensure success with your dual ISH-IHC assay. First, all the dual ISH-IHC protocols require some optimization. In general, it is recommended to combine a working RNAscope protocol with a working IHC protocol. It is also advisable to perform ISH first, followed by IHC. Also, it is advisable to optimize IHC assay separately using RNAscope pretreatment reagents to ensure that the protein can still be detected following the RNAscope pretreatment. Lastly, dual ISH-IHC assay works better for highly expressed proteins due to protease treatment that is used during the ISH protocol. We do have technical notes for this application on our web page. Here is an example of RNAscope Singleplex red assay to detect PDL1 probe with subsequent IHC staining in blue for CD45 protein by IHC. Here is an example of dual RNAscope assay with two markers followed by immunofluorescence staining. RNAscope markers are shown in green and red, while antibody staining is shown in white for Sci-Fi using the Multiplex Fluorescent V2 assay. Another area of RNAscope application is validation of RNA-seq results. Authors have used RNAscope-ish to confirm the identity of the neuronal subtypes identified using single nucleus RNA-seq. This paper demonstrates a robust and scalable method for identifying and categorizing single nuclear transcriptomes. Now we'll quickly switch gears to discuss BaseScope assay briefly. BaseScope is a next generation in situ hybridization assay. It is based on similar technology of RNAscope, but there are several major differences. First, a standard RNAscope probe contains 20 ZZ pair, which targets 1 kb region of the target sequence, whereas BaseScope can detect signal with as low as 1 ZZ pair probe. Both RNAscope and BaseScope are capable of detecting single molecule, meaning that single dot represents one single RNA molecule or target. RNAscope is a robust assay to detect gene expression of most RNA targets, whereas BaseScope is specifically designed for detection of exon junctions, short target sequences, which we'll look at in the next couple of slides. RNAscope is able to detect up to 3 targets, whereas BaseScope, we can detect 1 to 2 targets. Detection with RNAscope can be chromogenic or fluorescent, whereas BaseScope is currently available as fast-red detection for Singleplex, which can be visualized as chromogenic or fluorescent detection, and Duplex, which can also be detected as fast-red and green color and visualized as chromogenic. Please note that BaseScope probes are designed differently, so they cannot be combined with RNAscope reagents and vice versa. The assay workflow for BaseScope is very similar to RNAscope. Based on our experience, BaseScope assay may require stronger pretreatment due to additional amplification steps required. As a note, do not mix the RNAscope and BaseScope reagents as they are not compatible with each other. This is a protocol overview of the Duplex chromogenic assay using FFPE samples. The workflow is similar to the Singleplex BaseScope with a few additional amplification steps. This assay can usually take about 11 hours and can be divided into 2 days. Listed here are a few of the key areas of BaseScope applications. Due to limited time, I will present just one example for BaseScope Singleplex and Duplex assays. Please refer to our previous webinars for details on BaseScope applications. One of the applications of BaseScope is detection of short sequences. For example, the CDR3 alpha and beta sequences from the Jurkat cell line differ from each other by only approximately 50 nucleotides. BaseScope probes were designed to target either CDR3 alpha or beta, and the sense probe was used as negative control. BaseScope Duplex assay has been used to detect the CRISPR-mediated nucleotide deletion. CRISPR-Cas9 was used to delete 49 nucleotides from the gene, resulting in the novel junction sequence labeled as edited. BaseScope 1ZZ probes were designed to target either the 49 nucleotides in the wild type or the edited novel junction sequence. The image on the right shows the staining of wild type biallelic or monoallelic hepatocytes. Now, as we are towards the end of the webinar, I would like to guide you to the web page where you can find the additional information on the RNAscope and the BaseScope assays. Please visit our website at acdbio.com. If you click on the Support tab, we have a wealth of information including troubleshooting guides, user manuals, tech notes, training videos for manual assays, as well as previously recorded webinars that cover a wide range of topics and applications. With that, I would like to thank everyone for attending today's webinar. In just a couple of more minutes, I'll post some of the most frequently asked questions. My colleagues are still on the line to help address your questions. Please feel free to continue to send your questions through. Again, as a reminder, this webinar will be recorded and available on the website as previously shown. In the event you were unable to get a question sent through the chat or question and answer feature, please find the contact information for technical support on this slide. A few questions that are frequently asked are such that, do I need to run every single AMP reagent when doing the fluorescence multiplex assay? Yes, you need to run each AMP reagent as it serves as building block for the amplification structure of your target mRNA. If any of the AMPs are missed, this may result in no signal. Next, what does the C1, C2, and C3 nomenclature mean? In order to detect multiple mRNAs simultaneously, multiple amplification trees must be built. A C1 probe has a specific semen tail sequence that will allow binding of the subsequent semen pre-amp and the labeled molecules. Similar is true for the C2 and the C3 probes. This slide shows some of the other frequently asked questions.